You've heard about the mysteries of the deep ocean, the darkness, the pressure. The creatures down there look like something ripped from nightmares, but here's what's really going on. The reason the deep ocean is packed with these bizarre animals isn't just adaptation, it's a complete redesign of what life can be. We're talking about fish with transparent skulls and eyes that burn 600 times brighter than human vision. Squid the size of school buses floating in zones where oxygen barely exists, and mating strategies so extreme they involve males physically melting into females and becoming permanent parasitic organs. This isn't evolution playing it safe. This is survival under conditions that would crush everything you know about biology. 6,000 meters down, 8,000 meters, pressures exceeding 1,000 atmospheres, temperatures hovering just above freezing, no sunlight, almost no oxygen in some zones. And yet life doesn't just survive, it thrives. January 2024, Schmidt Ocean Institute launched an expedition off Chile's coast. Lead scientist Javier Solanes from Universidad Católica del Norte took the research vessel Falkor 2 into underwater mountain ranges nobody had properly explored. They used remotely operated vehicles and multi-beam sonar to scan seamounts at depths reaching 4,500 meters. What they found blew past every expectation. 100 new species, documented in less than 1 month. Deep-sea corals, glass sponges, squat lobsters with spiky shells and beady eyes. Cactus urchins where only one species was known before. Translucent glass squids. And ancient corals likely thousands of years old. If you value documented marine biology and deep ocean history like this, subscribe. It's the best way to support our work. Also comment from which city you're hearing this from. This script is researched and factual using sources from the Biodiversity Data Journal Volume 13, published November 2nd, 2025, by the Ocean Species Discoveries Initiative, reporting 14 new species from depths exceeding 6,000 meters, including carnivorous bivalves and parasitic isopods with popcorn-like structures. And from Conservation International's Deep Sea Expedition reports published March 4th, 2024, documenting over 100 species from the Salas y Gomez and Nazca ridges, where researchers found new species every few meters, according to lead scientist Aaron Easton. Now back to the information. Aaron Easton said it flat out in March 2024. It was almost baffling to discover so many new creatures. In some areas, we were finding species we'd never encountered before every few meters. The Salas y Gomez and Nazca ridges stretch 3,000 kilometers across the southeastern Pacific, longer than the Himalayas. These underwater mountains create unique ecosystems. Rotating currents carry cold water from the south and warm water from the north, high nutrient levels. Species living on nearby seamounts are completely distinct from each other. Each mountain functions like its own isolated world, and over 70% of these ridges sit in international waters, the high seas, beyond any single country's jurisdiction. May 9th, 2025. Ocean Census announced 866 new marine species discovered. The Nippon Foundation Nekton. Ocean Census ran the world's largest collaborative effort to document ocean life. Among the discoveries, a new guitar shark species found off Mozambique and Tanzania at 200-meter depths. Shark expert David Ebert identified it. Only the 38th known guitar shark species worldwide. 2/3 of guitar sharks are threatened. Marine gastropods discovered off New Caledonia and Vanuatu between 200 and 500 meters deep. These turrid gastropods possess venomous harpoons used to catch prey. Their peptides show potential for pain relief and cancer treatment. One drug for chronic pain was developed from a related snail family. August 16th, 2024. NOAA. Ocean Exploration published findings on benthic comb jellies. Scientists Nicholas Bezio and Alan Collins from the Smithsonian studied samples from 1928, 2018 and 2022. They used micro-CT scanning, 3-dimensional X-ray technology on century-old preserved specimens and fresh samples. The deep-sea benthic ctenophore genus Chalfiella hadn't been sampled since 1928, 90 years. Then NOAA expeditions in 2018 and 2022 hauled up living specimens. The samples matched closely. This comb jelly probably plays a role in far more deep ocean ecosystems than anyone knew. October 2nd, 2024. Schmidt Ocean Institute discovered a new seamount off Chile's coast during a 28-day expedition. The mountain rises 3,100 meters from the seafloor, nearly 2 miles, located 1,450 kilometers offshore. They created detailed 3-dimensional maps using advanced multi-beam sonar, then sent down remotely operated vehicles. What they filmed stunned everyone. Flying spaghetti monsters, Casper octopuses, a fish using fins like hands to walk across the seafloor, gardens of ancient corals, fields of glass sponges covering 800 square meters. the first ever footage of a live Promicoteuthis squid. This genus is so rare, only 3 species have been described mostly from dead specimens collected in the late 1800s. Here's where it gets strange. The pressure: 1 atmosphere at sea level, 10 atmospheres per 100 meters of depth. At 6,000 meters, you're dealing with 600 atmospheres. At 8,000 meters, 800 atmospheres. The Mariana Trench hits nearly 11,000 meters, 1,100 atmospheres. Every square centimeter of your body would experience force equivalent to 100 kilograms. That's the weight of 1,600 elephants standing on your head if you're a snailfish at 8,000 meters. Most organisms would be crushed instantly. Proteins denature. Cell membranes collapse. Bones shatter. But deep-sea creatures have solved this. April 15th, 2019, Nature Ecology and Evolution published genome analysis of Pseudoliparis swiri, the Mariana snailfish, caught at depths from 60,198 to 8076 meters in the Mariana Trench, the deepest fish ever caught with confirmed depth data. Scientists led by Shunping, he from the Chinese Academy of Sciences, sequenced its entire genome, then compared it to Tanaka's snailfish, a shallow water relative living in tide pools. The 2 species split from a common ancestor 20 million years ago. The Mariana snailfish has no gas-filled swim bladder. Gas compresses under pressure. Instead, its body cavity and tissues are filled with ammonium chloride solution, less dense than seawater. This creates neutral buoyancy without using gas. The bones aren't really bones. A gene critical for hardening bones. The bone GLA protein gene has a frameshift mutation causing early termination of cartilage calcification. The skeleton stays soft, gelatinous, flexible under crushing pressure. The skull doesn't close completely. Transparent, unpigmented skin. Scales so thin they're barely there. An inflated stomach to process the tiny crustaceans it eats. Hundreds of them shaped like roly-poly pill bugs found in their bellies when scientists dissected specimens. Cell membranes need to stay flexible at depth. High pressure makes membranes rigid and impenetrable. The Hadal snailfish solve this by increasing unsaturated fatty acids in cell membranes while reducing cholesterol and phosphatidylethanolamin. Groups of genes involved in fatty acid metabolism have expanded. The fish also produces trimethylamine N oxide, TMAO. This organic compound helps proteins maintain their shape under extreme pressure, prevents them from folding incorrectly. But here's the catch. Between 8,200 and 8,400 meters, there's a theoretical maximum depth for fish. Below that depth, fish can't accumulate enough TMAO to keep protein stable. Fish might tolerate slightly deeper for short periods, but would need to return upward. This creates a hard biological limit. June 20th, 2023. Scientists from eLife published findings showing the Mariana snailfish has multiple copies of 2 key genes. The gene CLDNJ, essential for forming otoliths, structures in the inner ear made of calcium carbonate. Normally, calcium carbonate can't efficiently accumulate beyond 4,000 meters, but the Hadal snailfish's otoliths are just as dense as shallow water relatives. Multiple copies of this gene solve the problem. The second gene, FTHL27, extra copies allow the fish to maintain auditory senses and withstand pressure. The fish has also lost many vision-related genes. Complete absence of light at depth means sight became useless. Some essential clock control genes remain, suggesting the fish maintains rhythmic cycles, but not based on sunlight. The loss of gene GPR27, which increases lipid mobilization, helps the fish survive starvation periods. April 12th, 2024. Researchers including Ariadna Mecho from Barcelona Supercomputing Center published findings from the Salas y Gomez Ridge, 160 species not previously known in the region, at least 50 suspected new to science. One discovery ranks among the deepest mesophotic corals in the world, extending Polynesian fauna distribution by several hundred kilometers. Fields of sponges and corals at depth, vulnerable habitats needing protection. April 2nd, 2023. Japanese and Australian researchers filmed a Pseudoliparis snailfish at 8,336 meters in the Izu Ogasawara Trench, 158 meters deeper than the previous record from 2017, close to the theoretical depth limit for all fish. Professor Alan Jameson, chief scientist, said they've spent over 15 years researching deep snailfish. In other trenches, like the Mariana, they were finding them creeping over 8,000 meters in fewer numbers. But around Japan, they're abundant. The fish filmed was extremely small, a juvenile swimming toward baited traps. While exact species remains unknown, it's identified as a Pseudoliparis snailfish. 2014. No AA and University of Hawaii. scientists aboard research vessel Falkor explored the Mariana Trench during the Hadal Ecosystem Studies expedition. They deployed 5 deep sea landers at targeted depths. They broke several records, deepest rock samples ever collected from the inner slope. These rocks represent some of the earliest volcanic eruptions of the Mariana Island arc. And they filmed a completely unknown variety of snailfish at 143 meters, white, translucent, broad wing like fins, eel like tail, slowly gliding over the bottom, different from any deep sea fish ever seen. In other trenches at this depth, there's typically one fish species, a snailfish. This one had a novel body form. Between 2014 and 2017, scientists collected 38 Pseudoliparis swirei specimens from the Mariana Trench, depths from 6,900 to 8,000 meters. Mackenzie Geringer, postdoctoral researcher at University of Washington's Friday Harbor Laboratories, led the study published November 30th, 2017. They dropped free-falling mackerel-baited traps equipped with cameras. The snailfish's small pink scaleless body measures up to 30 centimeters, about twice as long as a cigar. It can withstand pressure equivalent to 1,600 elephants standing on its head, named Pseudoliparis swirei after Herbert Swire, first navigating sub-lieutenant on HMS Challenger. The 1872 to 1876 circumnavigation discovered the Challenger Deep, habitat of the Mariana snailfish. Females carry unusually large eggs, up to 9.4 millimeters in diameter. Each female has up to 23 mature eggs larger than 5 millimeters, alternating with about 850 immature small eggs. No structures indicating embryo development were found, even in largest eggs. Evidence suggests larvae spend time in open water at relatively shallow depths. Less than 1,000 meters. Now the eyes. Giant squid, Architeuthis ducks, lives in deep ocean from 300 to 1,000 meters, sometimes deeper. The eyes measure up to 27 centimeters in diameter, 11 inches, pupil 9 centimeters, 3.5 inches. Only extinct ichthyosaurs had larger eyes. These massive eyes detect faint light, including bioluminescence in the deep dark. Large eyes gather more photons, critical when light is scarce. The giant squid probably can't see color, but can discern small differences in tone. Important in low light. The colossal squid Mesonychoteuthis hamiltoni has eyes the same size, up to 27 centimeters, among the largest in the animal kingdom, found at depths of 1,000 to 2,200 meters in the Southern Ocean, surrounding Antarctica. Temperatures just above freezing. The colossal squid reaches up to 14 meters, but is much heavier than the giant squid, more robust body. While giant squids reach 13 meters and win on length, the colossal squid has massive body mass, over 450 kilograms. Sharp hooks on tentacles, rare adaptation among squid, aids in capturing prey. September 20th, 2025, research published on how giant squid survive extreme pressure. They lack gas-filled organs, achieve neutral buoyancy through ammonium chloride solution throughout their bodies, less dense than seawater. Their tissues are soft and gelatinous, the mantle especially. No rigid air-filled structures or dense bone, largely incompressible. At cellular level, they produce specialized proteins and organic osmolytes, including trimethylamine N oxide. These molecules stabilize cellular proteins and enzymes, preventing them from denaturing under high pressure. This gives squid their distinctive fishy smell. The deeper the species lives, the more TMAO they have, the more they smell. Bioluminescence. Over 90% of organisms in the mesopelagic zone can produce light. The chemistry involves luciferin and luciferase enzyme with oxygen as catalyst. Different species have evolved unique ways to house and control light-producing chemicals. Specialized light organs called photophores. Some form symbiotic relationships with bioluminescent bacteria. The light serves multiple functions: communication, finding mates, hunting, defense. February 11th, 2025. Nature Communications. Biology published research on deep-sea shrimp visual evolution. Bioluminescent deep-sea shrimp in superfamily Oplophoroidea show higher visual opsin diversity among species migrating to shallower waters with variable light conditions. Species that evolved photophores show evidence of adaptive visual evolution. Positive selection for putative mid-wavelength. Sensitive opsin facilitates light source discrimination. These shrimp migrate vertically, move from deep waters to shallower depths at night to feed under cover of darkness. Return to deep waters during day for protection. Migration strength varies greatly, changing the light environment they experience. Visual systems face unique selective pressures. Distinguish vertical changes in ambient light during migration. Tell difference between bioluminescent light sources. Congener versus predator. Counter-illumination. common defensive strategy. Species like Hawaiian bobtail squid use downward-facing photophores to match dim light filtering from above, effectively invisible to predators lurking below. Deep-sea dragonfish, females modulate light patterns indicating reproductive status. Males respond with distinct signals. Bioluminescent courtship displays represent millions of years of evolution, adapted to finding mates in the largest dark space on Earth. The anglerfish, serratioid anglerfishes, reproduce through sexual parasitism. May the 23rd, 2024. Yale researchers published in Current Biology. Chase Brownstein and Thomas Neer examined how sexual parasitism works with other traits influencing diversification. These fish inhabit the bathypelagic zone, 1,000 to 4,000 meters depth, pitch dark, matte black bodies. Bioluminescent lures to attract prey. Large tooth-filled mouths, they drift in the largest, darkest, most sparsely occupied habitat on Earth. Prey and mates spread thinly across vast abyss. Sexual parasitism. Tiny males attached to much larger females. Some species, the males bite, females then release after mating. Others, the male permanently fuses. Obligate parasitism. The male's head dissolves into the female. Circulatory systems merge. He transforms into permanent sperm-producing sexual organ. Female Photocorinus spiniceps can reach 77 centimeters. Males 6.2 to 7.3 millimeters at maturity. Once claimed, smallest vertebrate known. Now often excluded from records because they're parasitic, not free-living. Cryptopsaurus quasi. One female found with 8 males attached. The genetics behind this, they needed to shed adaptive immunity, the system of specialized immune cells and antibodies that attack pathogens. Otherwise, female bodies would reject parasitic males. Researchers reconstructed evolutionary history of key genes involved in adaptive immunity. Multiple groups of deep-sea anglerfishes convergently degenerated their adaptive immunity, enabling sexual parasitism. This degradation happened as anglerfishes moved into deep sea. The transition from benthic ocean floor-dwelling ancestors to pelagic open ocean habitat occurred during major global warming 50 to 35 million years ago. High temperatures drove many deep-sea organisms to extinction. The midnight zone became prime real estate. Females grew larger. Males shrank to parasite size. Immune systems evolved to permit long-term flesh melting. Glowing lures evolved. Twitching appendage on forehead. Modified dorsal fin ray. The esca at tip contains symbiotic bacteria emitting blue-green light, irresistible to prey in darkness. Female controls intensity and can create pulsing patterns. This hunting strategy is effective because anglerfish can sit motionless and wait. Low energy expenditure. The lure serves double duty. Also attracts mates. The vampire squid, Vampiroteuthis infernalis, vampire squid from hell. Small cephalopod found throughout temperate and tropical oceans. Extreme deep sea conditions, 600 to 900 meters or more, sometimes reaching 3,000 meters. Within this region is the oxygen minimum zone, OMZ. Saturation of oxygen too low to support aerobic metabolism in most complex organisms. The vampire squid is the only cephalopod able to live its entire life cycle in the minimum zone. Oxygen saturations as low as 3%. November 22nd, 2012. Proceedings of the Royal Society B published findings from Monterey Bay Aquarium Research Institute. Henk John Hoving and Bruce Robison. 20 years of in situ observations. Laboratory studies of live specimens. Analysis of preserved material. Vampiroteuthis off central and southern California and both sides of Baja California Peninsula is primarily a detritivore. Takes advantage of OMZ, predation pressure less than surrounding mesopelagic layers, because respiratory constraints from diminished oxygen keep predators out. The vampire squid eats marine snow, dead animals, rotting materials, feces, mucus, dead plants, gelatinous zooplankton remains, discarded larvation houses, crustacean remains and molts, diatoms, fecal pellets, uses 2 retractile filaments to capture food. These filaments are between first 2 pairs of arms on dorsal side, thin and sticky. Collect drifting bits of organic material sinking from waters above. When filament contacts food, the squid retrieves it along arm tips to mouth. Feeding behavior, unlike any other cephalopod. February 18th, 2021, Communications Biology published fossil evidence, Nicroteuthis hungarica from Oligocene of Central Paratethys, first post-Mesozoic vampire squid. A 120 million year gap in fossil record separated living Vampiroteuthis infernalis from early Cretaceous ancestors. This fossil links them, assigned to family Vampiroteuthidea. Micropaleontological and geochemical analyses show it inhabited bathyal environments with bottom water, anoxia, and high primary productivity. Vampire squids adapted to bathyal oxygen, depleted habitats, at least since Oligocene. Cretaceous and early Cenozoic oxygen. Minimum zones likely triggered their deep sea specialization. The vampire squid reaches maximum 30 centimeters total length, 15 centimeter gelatinous body. Color varies from velvety jet black to pale reddish, depending on location and lighting. Webbing connects 8 arms, each lined with rows of fleshy spines called cirri. Inner side of this cloak is black, 2 long retractile filaments, eyes proportionally largest of any animal, up to 2.5 centimeters diameter in a 15 centimeter body. Blue blood. Hemocyanin binds and transports oxygen more efficiently than other cephalopods. Gills possess especially large surface area. Mass-specific metabolic rate lowest of all deep-sea cephalopods. Weak musculature, greatly reduced shell, maintains agility and buoyancy with sophisticated statocysts and ammonium-rich gelatinous tissues closely matching seawater density. Red coloration, unusual for deep sea. Red light doesn't reach deep ocean. Red objects appear black, blending into surroundings. Red pigmentation, more energy, efficient to produce than other colors. If disturbed, the vampire squid curls arms up outward, wrapping them around body, turning inside out, exposing spiny projections. If highly agitated, ejects sticky cloud of bioluminescent mucus containing innumerable orbs of blue light from arm tips. No ink sacs, different defense strategy. If you value documented marine adaptations and deep sea ecology like this, subscribe. Comment what fascinates you most about these creatures. This script uses peer-reviewed research from the Proceedings of the Royal Society, B 2012 study on vampire squid detritivory, published by Hoving and Robeson, showing extensive ROV observations and laboratory experiments. And from Current Biology, May 2024, publication on anglerfish sexual parasitism by Brownstein and colleagues demonstrating genomic analysis of 975 ultra-conserved elements from 230 species, revealing immune system degradation, enabling male-female fusion. Back to the depths. Living at extreme depths requires extreme metabolism. The Hodel snailfish at 7,000 meters has enlarged liver with unique lipid and protein metabolism. Research from Institute of Deep Sea Science and Engineering Chinese Academy of Sciences showed the fish increases lipid reserves, including cholesterol esters and triacylglycerols, to withstand prolonged food scarcity, elevates coenzyme Q and ATPase, enhancing energy production efficiency. Antioxidant adaptations include increasing ratio of monounsaturated fatty acids, enhancing levels of transferrin and heat shock proteins, reduces risk of lipid peroxidation. The metabolism of deep creatures drops dramatically. Less food available, colder temperatures. No need for speed when prey is scarce. The vampire squid drifts slowly, deploying filaments waiting for marine snow. Anglerfish hover motionless, lure dangling. Giant squid likely spend much time suspended in water column. When they do move, it's jet propulsion, suck water into funnel and mantle. Pointing. Force it back out. More water taken in faster they go. This action also passes water through gills, gathering oxygen. Abyssal gigantism, phenomenon where certain deep sea creatures exhibit remarkable growth due to extreme cold conditions. Giant squid, colossal squid. Both reaching massive sizes. Japanese spider crab, leg span up to 3.8 meters, giant isopods up to 50 centimeters long. Several factors drive this. Kleiber's rule: larger animals are more energy efficient. Metabolic rate doesn't increase proportionally with body mass. In food, scarce environment being large and slow is advantage. Buoyancy. In water reduces energy cost of supporting large body. Cold temperatures slow metabolism further. Larger body size helps retain heat in frigid water. Reduces relative surface area, minimizing heat loss. The oxygen temperature hypothesis. Polar gigantism. Cold water holds more dissolved oxygen, allows larger body sizes. Arctic and Antarctic waters host giant sea spiders, giant amphipods, Antarctic giant isopod. May 24th, 2024, Arctic Deep Expedition by Ocean Census and University of Tromsø. Species Discovery Workshop, October 2024. Red-colored creatures, abundant even at extreme depths thousands of meters below sunlight. Red pigmentation, energy efficient. Acts as camouflage since red light absorbed by water, making red creatures appear black. Discoveries never stop. July 17th, 2025, Ocean Census documented 9 incredible Arctic deep sea animals, red anemones, armored isopods with scavenging behaviors. Each organism plays a critical role in maintaining delicate balance of life in Arctic deep ocean. November 12th, 2024, Monterey Bay. Aquarium Research Institute discovered new species of sea slug in Midnight Zone, 1,000 to 4,000 meters below surface. Chinese researchers, November 2024, published in Yale E360, discovered chemosynthetic life at deepest point ever recorded. Using deep sea vessel, Fenduje, they explored depths over 9,000 meters. Abundant wildlife communities. Fields of marine tube worms with white snails, symbiotic relationship with chemosynthetic bacteria living in worm bodies. Bacteria provide nutrients. Worms provide stable habitat. White centipede-like worms in genus Macellecephaloids. Sea cucumbers. Various clams alongside anemones. Unlike ecosystems feeding on organic material falling from above these communities, sustained by methane produced by microbes buried under seafloor, suggests wildlife communities more common in extremely deep trenches than previously thought. Mengrandu, researcher at Institute of Deep Sea Science and Engineering, said, presence of chemosynthetic ecosystems challenges long-standing assumptions about life's potential at extreme depths. The biology gets stranger under pressure. Proteins need specific three-dimensional shapes to function. Pressure distorts molecular structures. Deep sea organisms counter this with pressure-adapted proteins. Some actually work more efficiently under high pressure than at surface. Cellular membranes must maintain fluidity. Lipid composition shifts. Higher proportion of unsaturated fats keeps membranes flexible. DNA repair mechanisms enhanced. Radiation from natural radioactivity higher at depth. Potassium-40 in seawater produces Cherenkov radiation from beta decay. Deep sea creatures developed robust DNA repair, osmoregulation, maintaining proper balance of salts and water in cells. Deep sea fish, blood similar salinity to seawater. Reduces osmotic stress. Ammonium chloride serves dual purpose. Buoyancy and osmotic balance. Some deep creatures produce organic osmolytes. Small molecules protecting proteins from pressure stress. Besides TMAO, there are others. Betaine, sarcosine. These compounds counteract pressure effects on proteins. Sensory systems adapt. Vision becomes less important as light disappears. Many deep fish have tiny eyes or none. Others, like giant squid, have enormous eyes maximizing photon capture for bioluminescence detection. Lateral line systems enhanced. Detect water movement vibrations, critical for finding prey and avoiding predators in darkness. Electroreception in some species. Detects electrical fields generated by muscle contractions of other animals. Chemoreception highly developed. Sense chemicals in water over long distances. Finding food, finding mates. Reproduction strategies shift. Low population density makes finding mates difficult. Some species become hermaphrodites, can change sex when needed. Anglerfish males parasitize females, solving mate location problem permanently. Deep sea octopuses brood eggs for years. 4.5 years recorded for Granelodon boropacifica, longest brooding period known. Female guards eggs entire time. Doesn't eat. Dies after eggs hatch. The giant squid reproduces only once in its approximately 5-year lifespan. Males don't use modified arm to transfer sperm like most squid. Sperm packet expelled from penis, sticking through funnel. Can be up to 7 feet long. As long as animal mantle, male injects sperm packets directly into female arms. Sperm could travel through arms to fertilize eggs internally, or arm injection triggers ovaries to release eggs bound with jelly held in arms. Sperm sense eggs nearby and migrate to fertilize. Lifespan varies dramatically. Small deep sea fish might live decades. Slow metabolism, cold temperatures, low food availability all extend. The lifespan, orange roughy, can live over 150 years. Greenland shark potentially over 400 years. Longest lived vertebrate. Deep sea corals live thousands of years. Some specimens over 4,000 years old. Growth rates incredibly slow, millimeters per year. Food webs in deep ocean fundamentally different. Surface ocean has phytoplankton base. Photosynthesis drives everything. Deep ocean mostly relies on marine snow, organic particles sinking from above, dead plankton, fecal pellets, mucus, detritus. This material sinks slowly, most consumed before reaching abyssal depths. Only about 5% of surface production reaches deep seafloor. Creates food scarcity. Opportunistic feeding strategies common. Many deep creatures can consume prey larger than themselves. Expandable stomachs, massive jaws. Anything encountered is potential meal. Hydrothermal vents create exception. Chemosynthetic bacteria use chemicals from vents as energy source. Hydrogen sulfide, methane. These bacteria form base of unique food web. Tube worms, giant clams, vent crabs, entire communities independent of sunlight. Cold seeps, similar. Methane or hydrogen sulfide seeping from seafloor supports chemosynthetic communities. Human impact reaching deep ocean. Bottom trawling destroys ancient coral gardens, takes over 1,000 years to grow, wiped out in single pass. Deep sea mining threatens seafloor ecosystems. Plastic pollution found even in Mariana Trench. In deepest fish, microplastics documented in vampire squid and other deep cephalopods from southwestern Atlantic. Climate change affects deep ocean, warming temperatures, changing ocean chemistry, deoxygenation, oxygen minimum zones expanding. This might favor species like vampire squid adapted to low oxygen, but could stress others. The High Seas Treaty, United Nations agreement finalized to protect international waters, creates framework for marine protected areas beyond national jurisdiction, critical for deep ocean. Most underwater mountains and ridges in high seas. Over 70% of Salas, I Gomez, and Nazca ridges outside protected waters. Implementation challenges remain. Needs formal adoption and ratification by at least 60 countries, but represents major step toward protecting deep ecosystems. Conservation efforts increasing, blue corridors proposed, connecting protected areas across ocean regions, allow species, migration, and genetic exchange. Ocean census accelerating species discovery. 866 new species announced May 2025. Discovery rate increasing with better technology. Remotely operated vehicles, autonomous underwater vehicles, advanced imaging, genetic analysis, environmental DNA sampling. These tools revealing ocean biodiversity faster than ever. The Schmidt Ocean Institute conducting ongoing expeditions. Research vessel Falkor 2 equipped with advanced multi-beam sonar. ROV Sebastian capable of 4,500 meter dives. High-definition cameras, sampling equipment, real-time video streaming. Public can watch discoveries as they happen. YouTube channels broadcasting live feeds, democratizing ocean exploration. 350 anglerfish species globally, over 300 snailfish species, 500 squid species estimated in ocean. Almost all squid in same taxonomic order as giant squid, called Oegopsina. Some surprisingly tiny, only about 2.5 centimeters. Others massive. Biodiversity in deep ocean likely exceeds surface ocean. Ocean covers 71% of Earth's surface. Deep ocean over 90% of biosphere by volume. Largest habitat on planet. Yet we know less about it than surface of Mars. Technology advancing rapidly. Deep sea cameras improving. Low light sensitivity crucial. Bioluminescence cameras capture light production. Pressure housings allow equipment to survive crushing depths. Battery technology extending mission duration. Acoustic systems map sea floor in unprecedented detail. Genetic sequencing getting cheaper, faster. Can analyze environmental DNA from water samples. Detect species without seeing them. Machine learning helps identify species in video footage. Process massive amounts of data quickly. The next frontiers, deeper trenches, more seamounts, under ice exploration in polar regions, longer duration observations, understanding behavior, not just anatomy. Tracking individual animals over time, migration patterns, feeding ecology, reproductive cycles. Most knowledge comes from dead specimens. Need more observations of living animals in natural habitat, but access difficult, expensive, dangerous. Submersibles advancing, Alvin upgraded. Can reach 6,500 meters. Limiting factor reached Challenger deep, full ocean depth capability. 10,925 meters. Private sector entering. OceanGate tragedy highlighted risks, but also showed growing interest in deep exploration. Billionaires funding ocean research. Different model than traditional government science funding. Pros and cons debated. The deep ocean holds answers to fundamental questions. Origin of life. Early Earth had reducing atmosphere. Deep sea vents provide similar chemistry. Extremophile bacteria there might resemble first life. Understanding pressure, adaptation could inform search for life on ocean worlds. Europa, Enceladus, Titan. If life exists under ice of these moons, it faces similar challenges. High pressure, no sunlight, limited resources. Studying Earth deep sea prepares us. Biotechnology applications emerging. Deep sea organisms produce unique compounds. Enzymes functioning at extreme temperatures, pressures. Antifreeze proteins, pressure-resistant proteins. Pharmaceutical potential. Cancer drugs from cone snails. Pain medications from deep snails. Bioluminescent proteins used in medical research. Deep sea sponges producing antibiotics. Chemical warfare between sessile organisms drives innovation. Humans harvest their arsenal. Climate regulation. Deep ocean absorbs carbon dioxide. Biological carbon pump. Organisms at surface consume CO2 through photosynthesis. Die. Sink. Carbon locked in deep sediments for millennia. Disrupting deep ecosystems disrupts carbon cycle. Mining, trawling, pollution, all threaten the service. Economic value difficult to quantify, but potentially enormous. avoiding climate catastrophe worth trillions. Mysteries remain. What exactly happens during reproduction in giant squid? No one has observed mating. How long do anglerfish males survive after attachment? Months, years, decades. What percentage of anglerfish females carry males? How many males per female typical? Vampire squid reproduction poorly understood. Eggs found free-floating but developmental stages unknown. Do larvae really spend time in shallow water? If so, how do they survive transition to deep oxygen minimum zone? Why do some trenches have more species than others? Mariana Trench, relatively low diversity. Kermadec, trench higher. What factors drive this? Geographic isolation, age of trench, food availability, ocean currents, temperature, oxygen levels, combinations of factors. Need comparative studies across multiple trenches. How will deep ocean respond to climate change? Warming affects oxygen solubility. Warmer water holds less oxygen. Oxygen minimum zones expanding. Does this favor oxygen-adapted species or stress ecosystems beyond tolerance? Acidification from CO2 absorption affects calcium carbonate structures, shells, skeletons, otoliths, deep-sea creatures with calcium structures vulnerable, but deep ocean already has high CO2. Variable carbonate chemistry. Some species might be pre-adapted. What lives in deepest trenches? Below fish limit, 8,400 meters down. Still 11,000 meters to reach bottom. Amphipods abundant. Giant amphipods. Swarms at 10,500 meters documented. But what else? Microbes certainly, probably unique species, worms, possibly other invertebrates. No one has comprehensively surveyed ultra-deep zones. Technical challenges immense. Equipment must withstand over 1,000 atmospheres. Sample collection difficult. Video recording possible but limited. Hadal zone named after Hades, Greek underworld. Appropriate given darkness and inaccessibility, extends from 6,000 to 11,000 meters. Mostly consists of ocean trenches, subduction zones where tectonic plates collide, deep V-shaped valleys, isolated from each other, allow speciation. Each trench potentially has unique species found nowhere else, makes conservation challenging. Protecting one trench doesn't protect others. The deep sea hosts, extreme specialists, organisms adapted to narrow niches, makes them vulnerable to disturbance. Recovery times measured in centuries or millennia. Slow growth, slow reproduction, low population density. Human activity could cause extinctions before species even discovered. Race between exploration and exploitation. Need to document and protect simultaneously. One more thing, the deep ocean isn't static. Currents flow. Water masses mix. Deep water forms at poles. Cold, dense water sinks, flows along ocean bottom toward equator. Takes centuries to complete circuit. This thermohaline circulation drives global climate. Deep ocean stores heat, releases it slowly. Changes in deep circulation could trigger abrupt climate shifts. Happened in past, could happen again. Understanding deep ocean, crucial for predicting Earth's future. The biological pump matters. Carbon fixed at surface sinks to depth. Removes CO2 from atmosphere. Without this pump, atmospheric CO2 would be much higher. Deep sea organisms part of this system. Bacteria decompose sinking material. Release nutrients. These eventually return to surface through upwelling. Fertilize surface productivity. Everything connects. 2024 saw record discoveries. 866 new species from ocean census. 100 from Chile seamounts. 14 from depths exceeding 6,000 meters. 50 from Salas Y. Gomez Ridge. One new snailfish record at 8,336 meters. Fossil vampire squid bridging 120 million year gap. Anglerfish immune system evolution explained. Deepest chemosynthetic communities documented. Each discovery raises new questions. The deep ocean is full of nightmares if you define nightmares as creatures, so alien they break everything you thought you knew about life. But here's what they really are. Solutions. Every bizarre adaptation solves specific problem. Pressure. Use soft bodies and pressure-resistant proteins. Darkness. Develop bioluminescence or enhanced non-visual senses. Low oxygen, slow metabolism, and efficient oxygen transport. Food scarcity. Opportunistic feeding and energy, efficient movement. Low population density. Extreme sexual strategies. These aren't monsters. They're masterpieces of evolution. 4 billion years of trial and error refined into organisms that thrive where nothing else can. And we're only beginning to understand them. 2025 continues the discoveries, new expeditions launching, more trenches to explore, more seamounts to survey, more species to document. Technology improves yearly, costs decrease, access increases. The deep ocean community growing, International collaboration accelerating, data sharing improving, genetic databases expanding. Every new genome sequenced reveals more about adaptation mechanisms. Every new species discovered fills gaps in the evolutionary tree. The reason the deep ocean is full of nightmares is because down there the rules change. What's impossible at surface is necessary at depth. Biology redesigns itself, pushes limits, explores possibilities we never imagined. Until we looked, until we sent cameras into the abyss, until we caught specimens and studied their genes, their chemistry, their structures. Now we know. Life doesn't just survive the deep ocean. It conquers it, thrives in it, diversifies across it, creates ecosystems as rich and complex as any rainforest, just hidden, just waiting. Just beneath the surface of the largest unexplored frontier remaining on Earth. 1901, first Hadal Zone fish caught. Abyssobrachula galathiigi at 8,370 meters. 2017. Pseudoliparus sweari officially described from 7,966 meters. 2023. New record at 8,336 meters. Progress spans over a century, but accelerating. What took 100 years to discover could be matched in next decade. Maybe sooner. The nightmares of the deep aren't slowing down, and neither are we.